Hi everyone, am I audible? Hello. Hi everyone, am I audible? So welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. So I have learned one line of Tamil, so I'm going to try that. No offenses. Okay, so Vanakkam. Yepidi Irukenga. I'm your biology educator Ambika Sharma. So that is going to be my starting line. Okay, the opening statement. Fine. So I've learned one line in Tamil and that is Yepidi Irukenga. It means how are you if I'm not wrong. Hannah, if I'm not wrong. Telugu, today my students are going to teach me Telugu. So tomorrow also I have one class at 10:30 a.m. So then I will use that Telugu line. And I am going to learn Kannada as well. Fine. So I will learn. So I am going to learn this statement only, that how are you? Hello, how are you? Fine. So welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and students, today we are going to start a new chapter, and you know that is your molecular basis of inheritance. So genetics, it is the most awaited topics for the NEET aspirants, right? In class 12th, you know that it is the most important unit that we cannot skip, and for the class 11 syllabus it is the physiology. So parallelly we are learning both, right? So we are already done with the principles of inheritance and variation. Today we are going to start molecular basis of inheritance. And let me tell you one thing, students, that soon I will take one shot of principles of inheritance and variation for your quick revision. Fine. So if you guys are having any doubt in that chapter, do attend that one shot. It will clear all your doubts. Fine. So this is what we are going to start today, and don't worry, right? In next class my opening statement will be in Telugu. Okay, so excited for the new chapter, students? Tell me. Today we are going to talk about the DNA, basics of DNA, all the bondings, right? Everything we are going to cover about the DNA. Okay, and you know what you have to do if you're new to our channel? You simply need to subscribe our channel, and here you people can see. Just have a look. This is the video that I have posted few days back. Okay, so it's the complete strategy video, bache. If you have seen it, very good. If not, please go for it, because here this is a kind of one-shot solution for all of you. Okay, this video is a one-shot solution for all of you, because bache, in this video I have mentioned the important chapters of physics as well, important chapters of chemistry as well, and biology is my forte, of course, that I will do. Fine. So do check it out. And if you want to join our batch, in which today I'm going to start chemical control and coordination, chemical control and integration. So you know what you have to do? In the description box, there will be the link for our batch. Here you can see the link for this chemistry mind map batch by your HSP sir. If you want, to revise chemistry, if your chemistry is not so good, this batch is perfect for you. And yes, Avengers batch also, you just need to click on any of the video in the description box. You will get the link. Click there and be the part of that batch, right? Be the part of that batch. So now let's start the class. Okay, one student is asking a question. What is the difference between the... Okay, see, when you are saying that someone, just say bacteria is exclusively heterotrophic. Exclusively means like all the bacteria there are heterotrophic, right? You know the meaning of heterotroph. They are dependent on others for their food, right? Exclusively heterotrophic. Like in the case of phylums also, when you talk about the phylum Echinodermata, we say na exclusively marine organisms are there. So when you are using the word exclusively marine, okay, yeah, exclusively marine means all the organisms of that particular phylum, they are living in the marine water. You are not going to find any organism there in the fresh water. Same way, if you're saying bacteria are exclusively heterotrophic, you are going to say that bacteria are not going to cook their own food, they are dependent on other, right, bache? And simply if you are saying bacteria are heterotrophs, in general you are saying, okay, bacteria, they're heterotrophs. But you know what is the correct thing, right? Bacteria, they are also autotrophic. You have the example of photosynthetic bacteria as well. Clear, bache Trisha? Okay. Now let's start the class. See, in the last chapter, in the genetics, if you are saying genetics and evolution, you include 3 chapters: principles of inheritance and variation, molecular basis of inheritance, and then the evolution. But if I'm saying simply genetics, so principles of inheritance and variation, then comes the molecular basis of inheritance, right, bache? Now, when we talk about the principles of inheritance and variation, right? When this is our chapter, principles of inheritance and variation, principles, certain rules which are responsible for the inheritance, right, which are responsible for that heredity, which are responsible for passing characters from one generation to another. But here you are talking about the molecular basis, right? Here you are not talking about the general rules, right? As per that rules, you know, the information passed from one generation to another, that you study in the principles of inheritance and variation. But when you talk about the molecular basis, here you are saying that which molecule will pass from one generation to another. We know simple thing that when characters, they pass from parents to offspring. Okay, when characters, they pass from parents to offsprings, we used to call it as heredity, right? What we used to call it? We used to call it as heredity, right, bache? The process of this transmission is known as inheritance. Simply when I'm saying that characters will pass from parents to offspring, that is heredity. But when I'm saying the process, right, of transmission of characters from parents to the next generation, then it is the inheritance. So here, right, so in the principles of inheritance and variation, that's what you study. Now we have to, we need to know that which thing will pass from one generation to another. Is it carbohydrate? Is it protein? Right? Is it lipid? Is it nucleic acid? What is it exactly? Right? And you know that after studying, right, after doing so many, after performing so many experiments, right, scientists, they got to know that it is the DNA which will pass from the one generation to another. So in this particular chapter, we talk about the molecules, right? Here again we are dealing with the heredity, but here we are talking about the molecular basis of heredity. Okay, that what exactly will pass from one generation to another. So that's what we have to discuss in this particular chapter, that is molecular basis of inheritance. So what is the plan for today? The plan for today is your DNA. Okay, it is your plan for today, and DNA stands for deoxyribonucleic acid. What is it? It is deoxyribonucleic acid. That's what your DNA is. Clear, bache? That is what your DNA is, and that's what we need to study. So do you know about them? Yep. Do you have any idea? Who are they? Anyone in the class? Yes. And please hit the like button, and if you're new to our channel, you know what you have to do. Just subscribe our channel. Tell me, anyone in the class? Array, James Watson, Francis Crick, right? James Watson, Francis Crick. Watson and Crick, they are. Okay, and you know that they explained the bDNA, they explained the DNA structure, but that is not true, right? Actually, Rosalind Franklin, you know about her? Rosalind Franklin, and this is, right, she performed the X-ray crystallography, X-ray diffraction, and she got the image of that DNA, right? It's just that before her, Watson and Crick, right, they got to know about her results and they explained the structure of the DNA. But actually she is behind this, right? If we know about the DNA structure, it's because of her. Okay, so this is the story that I'm going to tell you, but not today. Today we are simply going to start with the DNA. Okay, so bache, when you talk about the DNA, see, these are the general things from your NCERT. So when you talk about the DNA, deoxyribonucleic acid, bache, first of all, Frederick Mischer. Okay, this is the name of the scientist. Okay, Frederick Mischer. In 1868, he was the one, right, who found DNA in pus cells, right? In the nucleus of pus cells. In the nucleus of pus cells, he found DNA, right? And he called it as nuclein. He didn't use the term DNA, right? For the very first time, DNA was identified by Friedrich Mischer in 1868 in the nucleus of pus cell, and he called it as nuclein. And later on, Altman, because of its acidic property, Altman named it as nucleic acid. Okay, Altman named it as nucleic acid. So this is the first thing that you need to understand. Clear, bache? So what are we going to start? We are going to start the nucleic acid. Fine. What are we going to start today? We are going to start the nucleic acid. Now, bache, in this chapter, okay, now I'm just going as per the flow given in NCERT. So now in this chapter, first of all, you will talk about the structure of DNA. Okay, in detail we will discuss that DNA is made up of what. Then slowly we talk about the search for the genetic material, right? That which experiments were conducted to figure it out. Okay, then how that information will be passed, what is happening exactly. This is what we have to cover here. So we have to start the story from the nucleic acid, and in the upcoming sessions you people will read that how they concluded that it is actually the DNA. Okay, as of now you know that, I know that it is the DNA which is the genetic material, but later on I will tell you about the experiments as well. So what are we going to cover? We are going to cover the nucleic acid. So if we are saying acid, it means it is having the acidic property. So we talk about two nucleic acids. One is your DNA, another is your RNA. If you have attended my biomolecule session, then you will understand it in a wonderful way, because we have covered all that things there in the biomolecules as well. And no doubt, even today also I will explain everything. But yes, do check out the biomolecule session after this class. Fine. So when it comes to the nucleic acid, do you know that they have high molecular weight, isn't it? What do they have? They have high molecular weight, and you know that nucleic acid is also a biomacromolecule. So its molecular weight is above 10,000 Dalton. So when you talk about the nucleic acid, these are the two things that we discuss, DNA and RNA. Actually, bache, it is the DNA which is our main genetic material. Okay, in this chapter we will talk about both DNA and RNA, but here it is the DNA which is our main genetic material. RNA is performing other duties, like to convey the information from DNA to to convey the information so that it can form protein. That job will be done by RNA. RNA will help to bring amino acids from the cytoplasm for the polypeptide formation. Fine. RNA will help to form the your ribosome as well. So that's how it works. And moreover, RNA is the genetic material. Okay, RNA is also the genetic material in some viruses, and we call such viruses as your retroviruses. Okay, where RNA is acting as the genetic material. For an example, HIV, human immunodeficiency virus, it is also the example of retrovirus. Clear, bache? Now whenever you talk about the nucleic acid, we have one thing here, right? We have one thing here, and that is they are actually the polymers of nucleotides, right? That's what we used to write whenever we talk about the nucleic acid. What are these nucleic acids? These nucleic acids are the polymers. They are the polymers of nucleotides. Chino bache, they are the polymers of nucleotides. Now what is the meaning of this line here? See, you know about glycogen, right? You know that glycogen is also known as animal starch, right? It is our stored food, isn't it? It is stored in our liver, it is stored in our muscles. So glycogen is the stored food, we know it very well. So this glycogen is also a polymer. It is a polysaccharide. It is made up of many glucose molecules, right? It is made up of many glucose molecules. So we used to call it as polysaccharide. That is, it is having many sugar molecules. It is having many glucose. So glycogen is a polymer of glucose. But now when you are talking about the nucleic acid, they are the polymers of nucleotides. Means they are formed by joining so many nucleotides, right? So many nucleotides are going to join, right? so many nucleotides are going to join and they'll be forming what? They are going to form what? They'll be forming the nucleic acids. Clear bache? Clear bache? So that's why we are saying that nucleic acid is the polymer of nucleotides, or we can say that nucleic acid is the polynucleotide chain. See, everything is written in NCERT. We need to know about each and every line here, right? So it is the polynucleotide chain. Is that clear? So they are the polymers of nucleotide, or you can say that nucleic acid is a polynucleotide chain, right? So before understanding, Priyanka, before understanding the nucleotides, right? So before understanding the DNA or RNA, you should know what is a nucleotide. So can you just tell me about it in the chat section? What is a nucleotide? Can you tell me about it? Yes, nucleotide, it is made up of three things. That is your nitrogenous base, sugar, and the phosphate. Nitrogenous base, sugar, and phosphate. These three things, they join, they form the nucleotides. So many nucleotides, they join, they form the nucleic acid. That's the story here. Clear? That's the story here. So first of all, what we need to understand? We need to understand the structure of your, yes, nucleotides. Okay? So we have to study three things. Nitrogenous bases. But it is even given in your class 11th biomolecule chapter. Sugar and phosphate. Actually, do you know what is there? Right? Which acid is there? Actually, you know what is there? This is a very important thing. It is H3PO, yes, 4. In the DNA or even in the RNA, this is what you are going to get. H3PO4. It was a question in the chemistry. I still remember when HSP sir was solving the paper, right? So for the DNA structure, they have used few words. Phosphoric acid or the phosphorus acid, something like that. That whether it is phosphoric, right, or it is phosphorus, if I'm not wrong. Isn't it? It was there now. Phosphoric or phosphorus, H3PO3 it is, or H3PO4 it is, isn't it? So when you talk about your DNA or even your RNA, so bache, that's the thing. See, H3PO4, so it is your phosphoric acid. It is your phosphoric acid which will form your DNA. Okay, it is your phosphoric acid sahi thi, which is going to form your DNA. It was a question in the chemistry, right? It was a question there in the chemistry, right? So this is what you need to study about the nucleotides: nitrogenous bases, sugar, and the phosphate. Nitrogenous bases, sugar, and the phosphate. But these nitrogenous bases, okay, see, one thing is very clear. We are using the word nitrogenous bases, okay? We are using the word nitrogenous bases, means these are the chemical compounds having nitrogen. Am I right? These are the chemical compounds having what? Having nitrogen. Isn't it? Isn't it? Yes or no? So, nitrogenous bases. What you have here? Nitrogenous bases. So, we are going to talk about the purines here. Okay. And we are going to talk about the pyrimidines here, right? Pyrimidines here. So just look at it. So see, you know that when it comes to our when it comes to the excretory material, okay, let's relate it with the human physiology a bit. When we talk about the excretory waste, what is excretion? It is the removal of nitrogenous waste. It is the removal of our, or you can say that simply, you can say that it is the removal of our metabolic waste. It is the removal of the metabolic waste. It is the removal of the nitrogenous waste. So when we talk about the excretion, where we are talking about the elimination, right, where we are talking about the elimination of the metabolic waste, we majorly use the word nitrogenous waste. Why? See, your nucleic acid is having nitrogen, your proteins are having nitrogen, your proteins are making basic framework of your body. Okay, so majorly in that waste, it is the nitrogenous waste that is present. Fine, bache. So nitrogenous bases, this is what we are starting. So here you are going to talk about the purines and you are going to talk about the pyrimidines. What are you going to discuss here? You are going to talk about the purines and you are going to talk about the pyrimidines here. So bache, for purines there is one trick, that is puga. For pyrimidine there is another trick, that is cut. So when it is the puga, GA, so purines are your adenine denoted by A and the guanine denoted by G. Okay, and when it is the pyrimidine, it's cut, it's cytosine, it's uracil, and then it is the thymine. It is cytosine, uracil and thymine. So cytosine denoted by C, uracil denoted by U, thymine denoted by T. Okay, thymine denoted by T, and here it is, purines are your adenine and guanine, right? Purines are your adenine and guanine, and here it is cytosine, uracil and thymine. This is what we need to study. These are your nitrogenous bases. Clear bache? These are your nitrogenous bases. Now just look at this. Look at this. Adenine, guanine. Adenine, guanine. See, adenine in the spellings. Can you see the 9 here? In the guanine also. Can you see the 9 here? And here I have given you the trick as well. Puga. Okay, 9. So it means purines are your 9-membered ring. And when you talk about the pyrimidines, it is a 6-membered ring. Fine, you need to remember this. Purines are 9-membered, right? It is actually both purines and pyrimidines. They are the compounds in which rings are present. Here, it is a 9-membered ring. Here, it is a 6-membered ring. And here, it's a 9-membered ring, and at position, see, you have to note down. So at position, yes, can you tell me? At 1379. Okay, at 1379, nitrogen is present. In the ring at position 1, 3 and 7 and 9, you will see nitrogen, right? And here it is a 6-membered ring. At 1 and 3, nitrogen is present. Okay, at 1 and 3, nitrogen is present. These are the rings, no? Right? Purine is also a ring. Pyrimidine is also a ring. It is a 9-membered ring. So in this 9-membered ring, at 4 places, nitrogen is present. At position 1, 3, 7 and 9. Pyrimidine, which is a 6-membered ring, here at 2 places, nitrogen is present. 1 and 3. That's what you need to remember. Okay? That's what you need to remember. These are your nitrogenous bases. These are your nitrogenous bases. So have a look. See, this is the ring form. So pyrimidine. Look at this. 1, okay, pyrimidine. Done, bache? Done. So here in this pyrimidine, this is position 1, nitrogen is present. This is position 3, nitrogen is present. So that's why it's a 6-membered ring now. 2 nitrogen, 4 carbon. 2 nitrogen, 4 carbon. 1, 3, right? 1 and 3. Now, when it is a purine, as I said, bache, it is your 9-membered ring. Okay, it is your 9-membered ring. Fine. So see this. Nitrogen 1, it's 3, it's 7, it's 9. So in this ring, at 4 places nitrogen is there. Here 1, 3, here 1, 3, 7, 9. Important question. Okay, it is an important question. Is that clear? And if you will see this ring, you will see alternate single and double bond, then single, double bond, then single, double bond, then single. Same for this side. Okay, okay, is that clear? Is that clear? Yes, bache. So this is what you need to remember. Okay, this is what you need to remember. So just give me 1 minute. Done? Okay. Done, bache. This is your purine ring. Am I right? So 1, 2, 3, 4, 5, 6, 7, 8, and finally 9. Yes, bache, and finally 9. Is that clear? Yes or no? Is that clear? Yes? Is that clear? And here you know that double bond, single bond, double bond, single bond, double bond. And here also the double bond is there. Okay, right now I'm just talking about the ring. Fine, right now I'm just talking about the ring. Can you see here? The double bond and the rings, nothing else. Nothing else. You know, you have to satisfy the valency. So if it is 1, 2, 3, so you can write 4 here. 1, 2, 3, 1, 2. So of course, H here. Okay, and 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3. So here also, bus, done. Right? So you have to satisfy the valency here, because you know that your carbon is what? Your carbon is the tetravalent. So see, that's how the structure is present. That's how, right? That's how they are making it. Now, why am I telling you this again and again? See, in the case of pyrimidine, if you look at the structure very carefully, can I say that here you can see 2 rings? Yes, it is also a previous year question, bache. Can I say that you can see the 2 rings here? Pyrimidine and imidazole ring is present here. Pyrimidine and imidazole ring, together they are making your purines, right? Together they are making your purines. Just have a look. It is actually the pyrimidine, na? 6-membered, isn't it? It is actually the pyrimidine, yes or no? Can you see that? Can you see that? The 6-membered ring having 2 nitrogen and 4 carbon. This is what? This is the pyrimidine ring. And when you look at this side, it is imidazole. What's that? It is imidazole, right? It's a 5-membered ring in which 2 nitrogen are present and 3 carbon are present. So purine is formed by 2 rings. One is your, purine is formed by 2 rings. One is your, yes, one is your pyrimidine and another is what? It is your imidazole ring. Okay, one is your pyrimidine, another is your imidazole ring. So pyrimidine, 6-membered, right, where you have 4 carbon plus 2N, and here it's a 5-membered ring where you have 2 nitrogen and 3 carbon. Am I right or wrong? Bache, tell me, am I right or wrong? So purines, they consist 2 rings. One is the pyrimidine and another is the imidazole ring. Okay, another is the imidazole ring. That's what you need to remember. Just say once there was a PYQ, they have mentioned this. Purine consists of, options were there, pyrimidine, imidazole, both, or no one, like this. So you should know about it. Fine, you should know about it. So you have adenine here and you have guanine here. Now, see, these structures are given in your NCERT, in your class 12th biomolecule, class 11th biomolecule, even here in your molecular basis of inheritance as well. So if you can remember the structure, that will be very good for you. If you cannot, it's okay, it's your choice, but that's my suggestion, that you should remember the structures as well. And I will give you one trick here. See, when you are saying adenine, right, when you are saying adenine, rest everything is going to be same. Fine, you will see 1, 3, 7, 9 again. Right, you are going to see 1, 3, 7 and 9 again. But when it is the adenine, at the 6th carbon, you will see that NH2 group is attached. So many times students, they ask me that, ma'am, how can you say that in the adenine you have 4 nitrogen, 1 more nitrogen is there. But bache, here when we are saying 1, 3, 7, 9, nitrogen is present at position 1, 3, 7, 9, we are talking about the ring. Okay, no doubt in adenine you have NH2 as well, right? You have NH2 as well, but it is outside the ring. It is attached to the ring. It is not the part of ring. In the ring you have 4 nitrogens only. So it is adenine. When it is guanine, this is my way to remember it. Okay, guanine, O, 1 more O is added. So no doubt it is having the same. NH2 group is there, but again, it is having the C double bond O as well, isn't it? It is having the O as well. So that's how I remember. Guanine, guanine, O, there is a sound of O. So see, this O is present here. So that's how you can remember. Let's say in the paper they have given you two structures. Out of the following, which one is adenine, which one is guanine? So if it is guanine, O is there, O is there. So double bond O is present, right? Right? So here in the adenine, NH2 at the 6th carbon, and here again your NH2 is there, but it is at 2nd carbon, and the 6th carbon is having O, C double bond O. So by looking at the structure, you can decide whether it is purine or pyrimidine, right? And if you have to decide adenine and guanine, I have given you the trick. Now focus on the pyrimidine here. So we have 3 pyrimidines, right? We have 3 pyrimidines. So it is cytosine, it is uracil, it is thymine, right? I'll add one more thing here. This is thymine. This is also thymine, na? This is also thymine. But bache, this thymine is your nitrogenous base, and this thymine is what? Yes, it is your vitamin B complex, isn't it? Isn't it? Vitamin B1, thymine. So do not get confused ever. Do not get confused here ever, right? Like same way you have ileum and ILIUM. It is also ileum. This is the part of small intestine. This is the part of your pelvic girdle. Okay. ILIUM is the part of pelvic girdle. ILEUM is the part of small intestine. So it is thymine, THYMINE. It is nitrogenous base. If it is THIAMINE, then it is the vitamin B1. Fine, it is the vitamin B1. These are also the things that you should, notice. Fine. So now pyrimidine ring. So you have cytosine, uracil and thymine, right? So as I said, please focus on these structures as well. Now let's compare uracil and the thymine. See, if you will compare uracil and the thymine, you will see everything is same, isn't it? Everything is same, isn't it? Like see, it is the thymine. This is your carbon, nitrogen 1, 2, 3, 4, 5 and 6. So when you compare uracil and thymine, everything is same, but in thymine at carbon number 5 you have one extra methyl group, right? You have one extra methyl group. When it is the thymine at carbon number 5, you will see to this carbon CH3 is attached, that is your methyl group is attached. Okay, so thymine is also known as 5-methyl uracil. So if in uracil you will add methyl group to the 5th carbon, you are going to get your thymine. Okay, you are going to get your thymine. Right now, if you are thinking that ma'am, why are you teaching chemistry? Hai na? Just directly come to the point. So let me tell you, these things are the PYQs. Okay, whatever I am emphasizing right now, these were the questions in your previous year papers. Okay, so please mark it, right? So observe such things in the structures here. So uracil, thymine is also known as 5-methyl uracil. Okay, okay. So this is about what? This is about the nitrogenous bases. So now what you have to remember here, that in the case of DNA, okay, in the case of DNA, you have the purines as well, you have the pyrimidines as well. So which purines are there in DNA? Of course, adenine, guanine. Which pyrimidines are there? See, Y. So it is going to be cytosine. It is going to be thymine. Fine, it is going to be cytosine, thymine, Y. That's how you can relate. And now, even RNA is also the nucleic acid, right? Your RNA is also the nucleic acid. So RNA is also having the purines. RNA is also having the pyrimidines. But in the case of RNA, students, you will get adenine and guanine, no doubt. But in the case of pyrimidine, it is going to be cytosine and uracil. Uracil, R is there in that uracil. Right, Navansh? R, Navansh, is it right? So R is there. Uracil, R, uracil, R. So it is an RNA. Right, it is an RNA. Again, a previous year question. Fine, again it is a previous year question. So that's about the nitrogenous bases. Now we will talk about the sugars. The sugars present in these nitrogenous bases. So we have two things here, bache. Deoxyribose sugar and your ribose sugar, right? deoxyribose sugar and your ribose sugar. This is what we have. So one thing that you need to keep in your mind here is that this sugar, right, these are the pentose sugar. But what is the meaning of pentose sugar? That it is having 5 carbons. It is having 5 carbons. Okay. Okay, so which sugar is there in a nucleotide? Your pentose sugar, means total number of carbons in the ring, they are 5, right? How many carbons are there? 5 carbons are there. So one is a deoxyribose, of course it is in DNA, and ribose, R. So of course it is an RNA. That is another difference in between DNA and RNA. One is, in the case of DNA, pyrimidine is cytosine and thymine. In the case of RNA, pyrimidine is cytosine and uracil. In the case of DNA, the sugar is deoxyribose. In the case of RNA, the sugar is ribose. That's what you need to remember. That's what you need to remember. Now, bache, these sugars are the monosaccharides. What are they? These sugars are the monosaccharides, right? Monosaccharide. Mono means single, right? Saccharide means sugar. Mono means single. Saccharide means sugar, single sugar, right? It's a single sugar. It's a biomicromolecule. Okay, if you have missed the classes of biomolecules, please attend it, right? That's my suggestion. Complete biomolecules, molecular basis of inheritance, and biotechnology all together, right? So once you finish the syllabus, biomolecules, molecular basis of inheritance, and biotechnology, complete it together. Okay. Okay, that will be good, because one chapter helps in the revision of another chapter. So this is the single sugar. And you know, for monosaccharide, there is a general formula, CnH2nON. How many of you are aware of it? CnH2nON. Is it the formula, or there is something else? I'm talking about the monosaccharides, right? Just in the case of alkanes also, in the case of alkynes also, we have one general formula, right? We have the general formula, right? Yes. So here also we have one general formula. So can you tell me, is it right or wrong? Is it right or wrong? CnH2nON. It's right now. You can also write it like this. Okay. So N is 5 here. N is 5 here. So it will be C5H10O5. Fine. C5H10O5. Are you getting my point? Deoxyribose and ribose both are monosaccharide sugar. So in the case of monosaccharides, we have one general formula. Like when we study alkanes, alkenes, alkynes, alkane single bond, alkene double bond, alkyne triple bond is there. So accordingly, their general formula, it varies, isn't it? In the case of monosaccharide, that general formula is your CnH2nON, or you can also write it like this, CnH2O whole N. Okay. Okay. So if ribose is a 5, it is a pentose. If it is having 5 carbon, so this is going to be the general formula, C5H10O5, C5H10O5. Now, which I will mention one point here. That's very important. This is this deoxyribose. See, it's deoxy, means without oxygen. Without oxygen. We write it like this, 2-deoxyribose, right? That's how we write it, 2-deoxyribose. Okay. Thanks, Navansh. 2-deoxyribose means at second position, oxygen is absent, without oxygen. Now compare both the structures. I already told you that these sugars are pentose sugar, and this ring is a furanose, right? This ring is a furanose having 1 oxygen, 2 carbons. 1 oxygen, 2 carbons. But this is a wonderful chapter to revise biomolecules as well. Can you see that? Can you see that? 1, 2, 3, 4. 4 carbon, 1 oxygen is there. Right? So, see, carbon number 1, OHH. It is deoxyribose without oxygen. Second carbon is having just H and H. Here, HOH, CH2OH, and H. Now, compare it with ribose. Compare it with ribose. Okay. So, see, second carbon is having OH now. OH. But here in ribose, look at this. It is having what? H. So, if you compare the structure of ribose and deoxyribose, everything is same. Okay. everything is same. Okay. Okay. Everything is same. The only difference is here at second carbon, one oxygen is missing. So ribose follow the general formula. Ribose follow the general formula. But here deoxyribose, it doesn't. So it is having C5H10O4. This is the formula here as well. So deoxyribose is an exception also, bache. It doesn't follow the general formula of monosaccharide. It does not follow the general formula of monosaccharides. Are you getting my point? It doesn't follow the general formula of monosaccharides. Is that clear? Thanks, Ajay. But Ajay, I have a doubt. Ajay, I have a doubt. Initially, why were you feeling that it is the waste of time? Right? I really want that. Criticism is always, I always used to welcome this criticism. It helps me in the improvement. Will you please tell me why initially you were feeling it is the waste of time? I want to know that. Right? So see, these points are important. Fine, these points are important. Okay, so don't just focus on just cramming whatever is written in NCERT. Understand NCERT. Understand NCERT. All these things are written in NCERT, not in molecular basis of inheritance, maybe in biomolecules or in your class 12 chemistry biomolecules, but they are mentioned in your book. They are the part of your syllabus. That's why I'm telling you. Okay, that's why I'm telling you. So that's how you have to compare. So nitrogenous base is done, sugar's done. The next thing is your phosphate. Okay, the next thing is your phosphate. So bache, I told you already that when it is the nucleic acid, this is what you have. Okay, this is what you have. So it's H3PO4 and it is the phosphoric acid. Okay, it is the phosphoric acid which is present in the DNA. So these are the three things that we need to study. Fine, these are the three things that we need to study. So now see, nucleotides. Now first of all, your nitrogenous base will form bond with sugar. See, that's how I remember. NS. So when they join, they will form nucleoside. Fine, they will form nucleoside. When these three join, they will form nucleotide. See, that's how I remember. In the paper, they used to ask you, what is the difference in between the nucleoside and the tide? You know my trick, right? N stands for nitrogenous base, S stands for sugar. So when nitrogenous base and sugar, they join, they form nucleoside. And it is N and T. T means three things are there. Three things are there. So nitrogenous base plus sugar plus phosphate. Nitrogenous base plus sugar plus phosphate. Now Priyanka, what you have to remember here, bacha, nitrogenous base and sugar, they join with the help of N-glycosidic bond. Right? They join with the help of N-glycosidic bond. And here, nitrogenous base plus sugar, they will join with N-glycosidic bond. And when your sugar and phosphate join, here there will be the phosphoester bond. Which bond will be there? Phosphoester bond. Okay. So nucleosides, how many bonds are there? One. Okay. Nucleotide, how many bonds are there? Two. One is N-glycosidic, another is your phosphoester. So kindly focus here, it is phosphoester, not phosphodiester. It is phosphoester, not phosphodiester. Okay, not phosphodiester. Yes, but in Navansh, deoxyribose is also a monosaccharide. It is just that it doesn't follow the formula, but still it is a monosaccharide. Clear? So that's what you need to remember. Now, bache, if you know this part now, if you know this part, and then the structure of the DNA, the bonding of DNA, we discussed the 3,5 phosphodiester bond. That will be easy for you. Okay, see, that's how I remember. Sugar. Which sugar it is? Is it deoxyribose or ribose? Is it deoxyribose or ribose? Is it deoxyribose or ribose? Yes. Uh-oh. Deoxyribose, right? So sugar is sweet. Sugar is sweet. It is present here. Here, just write down your this part. This is your phosphoric acid, and here you have to write down either purine or pyrimidine. Let's say Array. What is it? Purine or pyrimidine? Purine or pyrimidine? It's your 9th nitrogen, 7th nitrogen, 8th carbon. Okay, so it's 1, 2, 3, 4, 5, and 6. Right? That's the structure. Yes or no? That is the structure. Yes or no? See, you can even compare. You can even compare. Right? Uh-oh. So this is the first one. Okay. This is the first one, second one, third one. That's how it is present. That's how it is present. First, second, third, fourth, fifth, sixth, seventh. See? First, second, third, fourth, fifth, sixth, seventh. I'm not talking about the double bonds and all. Chalo, theek hai. Let me draw that as well. Fine. Fine, fine, fine. That's the structure. Right? That's the structure. Now, bache, sugar is sweet. Right? We all love sweet people. Hai na? Hai na? We all love sweet people in our group. Right? You don't want a person who's always chaotic and always aggressive. Obviously, you want sweet people. Right? So, when you're talking about the nucleotide, see, here I have explained you one thing, that nitrogenous base and sugar will also join. You are going to use the word nucleoside. These three will join. You will use the word nucleotide. Now, I'm showing you the bonding here. Okay, what am I trying to show you here? Bonding. So this is how you have to put the sugar. Sugar, this is the sugar. Sugar is sweet. Even the phosphoric acid wants, phosphoric acid is going to form the bond with sugar. Even your purine is going to form. Even your nitrogenous base is going to form the bond with the sugar. That's what I'm trying to tell you here. Now, when it comes to the bonding with this phosphoric acid, fifth carbon of the sugar will participate. Jesse, let's say you are handling two projects, right? You are handling two projects. Okay. Okay, it is something like that. So sugar needs to get attached to the phosphoric acid. So its fifth carbon is involved in the bond formation. Here, this sugar will also join with this nitrogenous base. So its first carbon is going to form the bond here, right? The first carbon is involved in the bonding here. This part clear? This part clear? Yes or no? Tell me, this part clear? Yes or no? So you have to form phosphoester bond. So see, don't you think that from this part water can get eliminated? This phosphate will join here. Don't you think that from this part the water can get eliminated and your phosphoric acid will join here? Yes or no? It can get joined here. Yes or no? And here, if I'm talking about this part, okay, if I am talking about this part, 1, 2, 3. So H2O. So you need to eliminate the water from this side also. So if from this part, if I will eliminate the water, don't you think that I can join that ring with the sugar as well? Don't you think that this ring, I have to join the purine with the ring as well? Isn't it? Isn't it? Yes or no? Tell me quickly. Yes or no? Of course, yes, right? I'm showing you the bonding here, nothing else. I'm just showing you the bonding here. Just look at this. Look at this. Here they are showing you the base. This base will join with the first carbon of the sugar. And this phosphoric acid is going to join with the fifth carbon of the sugar. Okay, it is going to join with the fifth carbon of the sugar. That's what we are trying to show you here. Okay, that's what we are trying to show you here. So why are we saying that it is a phosphoester bond? Right? Why are we saying it's a phosphoester bond? First of all, tell me what is What is that ester bond, right? This now, C double bond O, and one more O is there, then it will get attached. This is what we study in lipids. But here, it is phosphate. It is like this. So that's why it is phosphoester bond. Priyanka, that's why it is phosphoester bond. So just look at this, 5th carbon. Here, the water will be removed. Here also, the water will be removed. So let me show you the attachment here so that you can compare. See, this is what I believe, that if you understand this portion, right, then you won't find it difficult. You are not going to find it difficult at all. So you need to throw out the water. H2O, O. Okay, H2O, wait. That's the story. Am I right? That's the story. Yes. And now here, am I right? Yes. So why are we saying it is the N-glycosidic bond? Because this nitrogen is involved in the bond formation here. And here it is the carbon, but this is the nitrogen which is involved in the bond formation here. Okay. Okay. So now see this. See this. This nitrogen is attached here to this ring. So now it is together, it is what? Now this is your nucleotide. What is it? It is your nucleotide. Is that clear? Is that clear? So bonding clear, carbon number clear. So bache, when it is a purine here, so its 9th nitrogen is involved in bond formation. Okay, its 9th nitrogen is involved in bond formation because it is the purine. Okay, so if nitrogenous base is your pyrimidine, then it is the first nitrogen. The bond will be in between C1 and N1. It is the first nitrogen, right? It is the first nitrogen of the pyrimidine. Nitrogen at first position is involved in the bond formation. This is also important to understand. Clear, bache Vidisha? Clear, bache Vidisha? So I'm repeating this portion again, right? Let's have a quick revision of it and then we'll go ahead. We are talking about the nucleotide. So if you want to understand the nucleotide, you should know about nitrogenous base, sugar and the phosphate. These three things we need to know. Nitrogenous base, sugar and the phosphate. Because if you know this bonding, if you know that how this bond will form, you are never going to make mistake in that 3-5- phosphodiester linkage. That's my advice. Now here I'm telling you about the nitrogenous bases first. I told you will be having purine, you will be having pyrimidine. So which purines do we have, guys? I want you people to respond here, right? Please type your answers in the chat section. I want you people to participate here in this discussion. So purines are adenine, guanine. Purines are adenine, guanine. Pyrimidines, cytosine, uracil and thymine. Now when you are talking about the DNA, right, this is also a difference. DNA is having adenine, guanine, cytosine, thymine. RNA is having adenine, guanine, cytosine, uracil. That's what we have in RNA and that's what we have in DNA. Now next is the sugar. Sugar is a pentose, bache Rithigna. Sugar is a pentose, 5 carbon containing sugar. The ring is furanose. So ribose sugar and deoxyribose, these are the two sugars. Deoxyribose is in DNA. Ribose is in RNA. This is the another difference. Deoxyribose without oxygen. So oxygen is absent at second position when you compare the structure of ribose and deoxyribose. This is what you observe. Okay. And then we talked about this phosphoric acid. Okay. H3PO4. So now here we are talking about the bonding. Fine, here we are talking about the bonding. So bache see, if it is adenine, adenine plus ribose, when they will attach, so they will form adenosine. They will form adenosine. So if it is adenine plus deoxyribose, then it will form deoxyadenosine. Then it will form deoxyadenosine. Are you getting my point? Then it will form deoxyadenosine. adenosine. Are you getting my point? Are you getting my point? Okay. Okay. So when the word is adenine, that's a nitrogenous base. If in your paper adenosine is written, then it is a nucleoside. So this can also be asked as a question in relation to that biomolecule. Now when it is the nucleotide, only one phosphate is attached. So can I say that, let's say if it is adenine plus ribose plus this phosphate, only one phosphate is attached? No. So can I say this? It's adenosine monophosphate. Can I say that it is adenosine monophosphate? Yes. Can I say that it is adenosine monophosphate? Of course I can. Of course I can. And it is also known as adenylic acid. So the naming should also be clear. Adenylic acid. So here if it will be deoxyribose sugar, then you will say deoxyadenosine monophosphate, deoxyadenylic acid. That's what you need to focus as well, Nandini. Okay. Okay. So same way, guanine plus ribose, guanosine, cytidine plus ribose, sorry, cytosine plus ribose, cytidine, uracil plus ribose, uridine, because there is no thymine in RNA, so it will be uracil plus ribose, uridine. Same for the DNA, guanosine plus deoxyribose sugar, it will be your deoxyguanosine, it will be your deoxythymidine, it will be your deoxycytidine. Okay, and when phosphate will be attached, you know how you need to change the naming there. Even that's what you need to remember. Fine. So this is the bonding. So sugar will be here. This is your phosphate. Okay, this is your nitrogenous base. So if nitrogenous base is purine, right? So carbon 1 of sugar, nitrogen 9 of sugar. So if it is the pyrimidine, carbon 1 of sugar and nitrogen 1 of pyrimidine. That's what you need to remember for the bonding. So do read it from the NCERT as well. So now I just said that when it comes to the DNA, it is a polynucleotide chain. DNA, RNA, they are polynucleotide chain. So see, polynucleotide means so many nucleotides will join together, yes or no? Polynucleotide chain means so many nucleotides, they will join together, yes or no? Yes or no? Right? Nucleotide 1, 2, 3, 4, 5, 6. These are the nucleotides. I need to join them. Just say if I was fixing so many glucose all together, I was making the glycosidic bond there. Now I have to join one nucleotide with another. So, bache, here, this bond will form. This is the bond that will form here, right? So now when it is a polynucleotide chain, okay, when it is a polynucleotide chain means when your nucleotides are joining together, how one nucleotide will join with another? With the help of this ester bond, phosphoester bond. So overall, when you are talking about the nucleotide chain, here you will see phospho diester bond. It is not the ester bond here, it is going to be diester bond. If it is a single nucleotide, then the bond is phosphoester bond. If it is a single nucleotide, the bond is phosphoester bond. But here it is what? It is the, yes bache, it is the phosphodiester bond. Okay, diester bond. Clear bache? That's how your polynucleotide chain will form. So whenever you are talking about the synthesis, means formation, whether it is RNA or it is DNA, this is a general rule that you need to remember, bache. This is a general rule that you need to remember, bache. Whenever you are talking about the synthesis, the formation of polynucleotide chain, it is always like this, 5' to 3' direction. What will be the direction? It will always be 5' to 3' direction. It will always be 5' to 3' direction, right? Like this one will be your 5' end having phosphate attached. This one will be your 3' end having OH, having hydroxyl. Here it is the phosphate. I will explain this part as well, but this is what you need to remember. Always synthesis, whether it is RNA or DNA, synthesis will always be in 5' to 3' direction. Let me show you this part. Okay. Is that clear? What is it? What is it? Is it a nucleoside or is it a nucleotide? The structure is what? Is it a nucleotide or is it a nucleoside? What is it? Quick. Is it a nucleotide or is it a nucleoside? What's that? It's a nucleotide, of course. So this is your carbon number 1, carbon number 2, carbon number 3, and which sugar it is? Is it the ribose sugar or the deoxyribose sugar? Is it the ribose sugar or is it the deoxyribose sugar? Which sugar it is? Deoxyribose. Excellent. Okay, so this is one nucleotide, this is the next nucleotide. Dorothy, that's the fact, right? It is your one nucleotide, it is your next nucleotide. It is your one nucleotide, it is your next nucleotide. Now see, here this is the sugar. Okay, this sugar, this OH, which is attached to the 3' carbon. This OH, which is attached to the 3' carbon here, it is going to, because we have to join one nucleotide to the another. See, this bonding is fine, CH2O, this is the phosphoester bond, this is the phosphoester bond, this is the N-glycosidic bond, this is the N-glycosidic bond. Now I have to join one nucleotide with the another. So the 3', right, this is how we write now, 1', second dash, third dash, or you also use the word prime, 1 prime, second prime, third prime. So this is a 3' carbon. This is a third carbon. This is a 3' carbon, third carbon. It is having this OH group. If you understand the structure, Pujita, then you can mark it very easily. It is the OH group. Now here you will see the bond formation. Here you will see the bond formation. Now from this part, your water will be eliminated. Right? From this part, your water will be eliminated. Can you see that? Can you see that? Yes. From this part now, the water will be eliminated. Actually, it is what? H2O now. Water is H2O. So, minus H2O will occur here again. So, now what is going to happen? See. That's how it will form. That's how it will form. Yes or no? See, OH was there. Here also OH. So you need to throw out water, minus H2O, right? Minus H2O. So that's what we'll get now. That's what we will get now. So see, when it is the phosphoester bond, that's how it will form. Right? If it is the phosphoester bond, now see, it is a phosphodiester bond. One is this side, one is present here. Right, this is P double bond O, and see, this O is attached to the carbon here. This is P double bond O, this O is again attached to the carbon here. So don't you think that this is the phosphodiester bond? Phosphodiester bond or phosphodiester linkage? Tell me, just give it a try. Give it a try. So see, so the first sugar, this is the first sugar, it's 3' carbon. This is the next sugar. This is the next sugar. See, carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. So next sugar mein 5th carbon. They are involved in the bond formation. Right? 3' OH, 5' this 5' carbon and this 3' carbon, they are involved in the bond formation with this phosphoric acid. Hai na? Yes or no? So that's why we used to say 3', 5' phosphodiester link. Right? This is 3-5- phosphodiester link. I think this is for the first time, right? Yeah, this is for the first time here on YouTube, when I'm drawing each and every structure and then I'm showing you that this is how water will get eliminated, that's how the bond will form. Otherwise, obviously, even I used to put the images and I used to explain that this is how the elimination is going on. But I am making each and everything so that you can understand. So please, please, please revise it after the class. So see, look at this part: sugar, nitrogenous base, phosphate. Dekho bache, we have enough time. Okay, we'll finish your syllabus. And here you know that if you have seen our lectures, you know that we teach you in detail. We are actually providing you the quality content. It's not like that we are saying that yes, we will provide you everything, and we are not. You know that in this batch we are delivering everything possible, and your NEET exam will not come beyond this part. Okay, now what you have to do, it doesn't matter that how good or how bad I'm teaching. If you are not revising, it is a waste for me. Okay, if you really appreciate this hard work, if you really feel that yes, they are providing us everything free of cost, then please start revising properly, start making notes properly, be consistent in the classes as well. Okay, attend the sessions regularly, and after class I just need your one comment in the comment section. Okay, one comment in the comment section. That's what I need from you people. Okay, so here you guys can see this is the nucleotide. Now this is the phosphoester bond. So when it is a polynucleotide chain, this sugar, 3- carbon of this sugar will attach to the next sugar at its 5th 5- carbon. That's why it's 3-5- phosphodiester linkage, and that's how it is. That's how it is, phosphodiester linkage, phosphodiester bond. And same will go for the next, for the next, for the next, and for the next. Now, why do we say that synthesis is always in 5' to 3' direction? Why do we say that synthesis is always in 5' to 3' direction? Simple as that. This is how you are going to put your nucleotide. Actually, it is the 3- carbon. Its OH is available for the bond formation with the next phosphate. So obviously that's why it is 3-5- phosphodiester linkage, and that's why the synthesis is in this direction. And moreover, always observe this thing, right? Always observe this thing. Now you are making the polynucleotide chain here. So can you see this part that 5- carbon is attached to the phosphate here? Can you see this part? Pujitha bache, can you see this part that 5- carbon is attached to the phosphate here? 5- carbon is attached to the phosphate here. Can you see this? Can you see this? 5- carbon is having the phosphate, and if you look at the 3- end, it is having the OH group. 3- end, it is having the OH group. That's what you need to remember. Always remember, whenever they will give you the polynucleotide chain in paper, if they are giving you the image, always make sure this is what you need to notice. 5- end is always having free phosphate group. 5- end is having free phosphate group. Free, why am I saying free phosphate group? Because it is not involved in any other bond formation, right? And when you are talking about the 3- end, it is having free OH group. Free OH group. Another MCQ. It is another MCQ that you need to remember, okay? That you need to remember. Wait, I'll show you the image. See this. So let's say if it will be a question in your final paper. So 5- end, if they are mentioning 5- end, always there will be the phosphate group, free phosphate group. And when it is the 3- end, here always you have to notice the free 3- hydroxyl. Clear bache? Clear bache? So see, nitrogenous base, sugar, phosphate. Nitrogenous base, sugar, phosphate. Phosphodiester bond. 3- carbon is involved, 5- carbon is involved. So that's how the bond formation is. That's how the bond formation will be there. See, look at this part. Okay, so that's what I need to explain you. So now the polynucleotide chain clear, the structure clear. I'll show you the images here. See. Now, even when you look at this part, can you see that in a polynucleotide chain, nitrogenous bases are somewhere in the center, and there is a sugar phosphate backbone, right? This is what we always used to say when we explain DNA structure, that there is sugar phosphate backbone. There is sugar phosphate backbone. Nitrogenous bases are there in the center, right? They're in the center, and there is always a sugar phosphate backbone. So see, sugar phosphate backbone it is, which is supporting this part, isn't it? Which is supporting this part, isn't it? So now just have a look, bache. This is your DNA double stranded, your RNA single stranded. So now that's the part that I will explain you. Okay, but as of now you can just have a look of this bonding. So that's how the bond formation will be there. Okay, that's how the bond formation will be there. So I will share the PDF, you will understand it, right? You can use this PDF as well. So see, this is sugar base phosphate, sugar base phosphate. So they will get attached to each other. So during DNA formation, actually it is the ligase which helps in this attachment. DNA ligase, you know, no, molecular glue helps in attachment. So see, that's how it is present. Any doubt? That's how it is present. Any doubt? Yes? Any doubt? So now you know the basics. Now let's revise it from NCERT and then let's go. Okay? Okay. So, bache, when it comes to the DNA structure, as I said, Watson and Crick, they explained the DNA structure. They got a Nobel Prize for that. But actually, your Rosalind Franklin, Maurice Wilkin, they worked on X-ray diffraction techniques, right? X-ray diffraction is also known as X-ray crystallography. They got some images and, you know, they used that images of Rosalind Franklin. Okay. And then Watson and Crick, they explained the structure. Actually, the credit, actually, no doubt, Watson and Crick, they are getting the Nobel Prize for that, but actually it is the work of Rosalind Franklin. It was the work of your Rosalind Franklin. She worked so hard and she got that image of DNA X-ray crystallography image, and then with the help of that Watson and Crick, they explained. So you will see, at that time even the sexism was there in the field of science as well, right? These Watson and Crick, they were jealous of Rosalind Franklin as well. Okay, the story is very interesting. She didn't get the recognition for her work in this case. She got many prizes for her other work, but yes, she is the main person behind, right? Okay, someday we'll tell you the story that what happened exactly. I'll make a separate video for that. Now focus here. So we are talking about the DNA. This line is clear now. It's a long polymer of deoxyribonucleotides. It's understood? It's understood? MCQ. Polymer of deoxynucleotide. Length of DNA is usually defined as number of nucleotides. If I have to say that this DNA is that much long, who's going to decide that? Number of nucleotides are going to decide that, right? Number of nucleotides are going to decide that. Each and every line written in this paragraph, it can come in your final exam as a question, right? So this is also your MCQ. Fine. So number of nucleotides or a pair of nucleotide referred to as a base pair. I will explain this part. Wait. This also is the characteristic of an organism. Like see, in the case of bacteriophage, this is, but you need to remember this data. A bacteriophage known as, what is it? It's 5 into 174. It is having 5386 nucleotides. Bacteriophage lambda is having 48,502 base pairs. E. coli has 4.6 into 10 raised to power 6 base pair. Everything is important here. And haploid content, haploid half. They are talking about the genome here. Haploid content of the human DNA is 3.3 into 10 raised to power 9 base pair. So if it will be the diploid content, it is going to be 6.6 into 10 raised to power 9 base pair. Right, 3.3 into 10 raised to power 9 base pair. So if it will be the diploid content, if you are talking about the diploid content, then it will be 6.6 into 10 raised to power 9 base pair. Clear, bache? Clear, bache? So how can you figure out the length of DNA? Right, it depends upon the number of nucleotides. Is that clear? It depends upon the number of nucleotides. Is that clear? So now just look at the structure of polynucleotide chain. And I know you know that, right? I know you understand this part, isn't it? You understand this part. So let's recapitulate the chemical structure of the chain. So you know that a nucleotide is having 3 components. We have covered this part, right? We have covered this part, right? Right? So it is having a nitrogenous base, of course. It is having a pentose sugar, ribose in RNA, deoxyribose in DNA, and it is having a phosphate group. Now you know this part as well, right? Now you know this part as well, isn't it? isn't it? So there are two types of nitrogenous bases, purines and pyrimidines. Purines and pyrimidines. Is that clear? Purines and pyrimidines. Is that clear? Okay. So cytosine is common for both DNA and RNA. Thymine is present in DNA. Uracil is present in RNA. Now see, each and every line of NCERT is decoded in this lecture. We have discussed each and every line of NCERT here. Clear, bache? So nitrogenous base is linked to the OH of 1-C pentose sugar through a N-glycosidic linkage to form nucleoside. See, even that's what I have explained. When I was explaining you the nucleoside, this is what I just said. This is what I just said. Nitrogenous base plus sugar will form your nucleoside, and N-glycosidic bond is here. And I told you the 5- carbon of sugar, and in the case of 1- carbon of sugar, and in the case of nitrogenous base, it can be 1N of pyrimidine, 9N of purine, right? 9N of purine, right? So see the naming also. So adenine plus ribose, adenosine. Adenine plus deoxyribose, deoxyadenosine. Now this part is clear or not, Trisha? So guanosine or deoxyguanosine, cytidine or deoxycytidine, uridine or deoxythymidine. So why like this? Uridine, because it is present in RNA. Thymine, deoxythymidine. Why? Because it is present in DNA, right? DNA. So when a phosphate group is linked to them, when a phosphate group is linked to the nucleoside, and you know that now your 5- carbon of sugar is involved. See, 5- carbon of sugar is involved now. Clear, bache? Clear, bache? So of a nucleoside through phosphoester linkage. See, so if it is a single nucleotide, which linkage will be there? Phosphoester. So do not get confused. Remember that image only, that sugar was in center. So 5- carbon of the sugar will form a bond with the phosphate group. 1- carbon of sugar of the 1- carbon of sugar will form the bond with the nitrogenous base. Is that clear? Right? Is that clear? So nucleotide, you know, so two nucleotides, when we have to link two nucleotides, they will be linked through 3-, 5- phosphodiester linkage to form a dinucleotide. So if it is a single nucleotide, which bond will be there? Phosphoester bond and N-glycosidic bond. If it is a dinucleotide, then it will be having N-glycosidic bond and phosphodiester bond. Diester bond. So in the polynucleotide chain, we have phosphodiester bond. We have phosphodiester bond. Okay. Now let's say I am giving you one question. Okay. Let's say in the NEET paper, I am asking you one question. I am asking you that, okay. Let's say this is the structure. Okay. This is the image you have in your paper. I'm asking you, I'm putting one arrow here, that what is A? Which bond is A here? What will you see? Which bond is A here? What will you see? What will you say? Right? If I'm putting here an arrow and I'm asking you in your final NEET examination, which bond is here? Which bond is there? Tell me. What will you say? Quickly answer it, everyone. Be quick. I have another class in 5 minutes, so please be quick. Excellent. It is phosphoester. Now, if I will mark this, what is A? What is A? What is A? Exactly, Nandini. Very good. Very good. So that's why I always used to say concept clarity is the most important thing. So more nucleotide can be joined with this, right? 3-, 5- phosphodiester linkage. So polymer thus formed at one end. See, at one end, and a free phosphate. See. So the polynucleotide chain at 5- end, it is having free phosphate moiety. Moiety means molecule, right? So that's why you are calling it as, you are simply referring it as 5- end. Similarly, at the end of the polymer, the sugar is having free OH group of the 3rd carbon. Clear, bache? That is your 3' end. And now you know that the backbone here is due to the sugar and phosphates. Right, sugar and phosphates. Any doubt here, bache? Tell me, any doubt here? So nitrogenous bases linked to the sugar moiety, they project from the backbone. Right? Right, they are present here in the middle. The sugar phosphate backbone is there. And when you are talking about the RNA, every nucleotide residue has an additional OH group present. They have an additional OH group present at 2' position in the ribose, of course. Ribose sugar is having extra 2' OH group. So in the RNA, it is the uracil at the place of thymine. Okay, so thymine is also known as 5-methyl uracil. 5-methyl uracil. So I told you this thing before that bache, each and every point is in NCERT. Okay, so see, DNA is an acidic substance. Why is it acidic? Because of the presence of your H3PO4, because of the presence of this phosphate, it is acidic, right? So it is present in the nucleus, was first identified by Frederick Mischer in 1869. Oh, I mentioned 1868, now it's 1869. Actually, in some books, 1868 is given. So it is 1869. So you can rectify that. So Frederick Mischer named it as nuclein. Frederick Mischer named it as nuclein. Fine, nuclein. However, due to technical limitation in isolating such polymer, the elucidation of structure remained elusive for a long period of time. Actually, see, he identified this nucleic acid in 1868, but the structure of DNA was given in 1953 by your Watson and Crick, right? And why such a long gap here? Because at that time, technique was not so advanced. Clear, bache? It was not so advanced. So in 1953, your Watson and Crick, based on, this is also a question, right? By using the X-ray diffraction, it is also known as X-ray crystallography. Clear? It is also known as X-ray crystallography. Right, so they produced by Morris Wilkin and Rosalind Franklin. They proposed a very simple but famous double helical model for the structure of DNA. So rest bache, I will discuss in next class, because after 5 to 10 minutes I have another class in your NEET Avengers batch, right? So that class will be there on our Unacademy platform. Clear? So whatever we have completed so far, please revise it. In the next class we will complete DNA structure and we'll be talking about the discoveries and all that points. So next class is also going to be very interesting. Fine. So I will share the PDF notes with you all, right? And okay, so I will share the PDF note in our official Telegram group. Don't worry about that. And tomorrow at same time, we will have another class, but do revise it and attend the classes regularly. And you know what you have to do, right? You have to write a comment for the class. You liked it or not, right? That's what you have to do. Just one comment. And I don't think that it is going to take much time. So right now 32 students are watching. So I'm expecting 32 comments. Okay, 32 comments. And yes, do check that strategy video, plan accordingly, and please decide, right, that which portion is strong, which portion is weak, how you need to, further you need to carry forward your, how are you going to revise further, in short. Okay. Sure. So that's it, that's all. So here, this is the video, right? I have posted it on 22nd of September. So do check this, the strategy to score 650 marks in 6 months. So everything is explained here in detail with a revision plan and with the physics, chemistry and the biology chapters. Fine. So take care. Bye-bye. Thank you so much for watching. So see you all. Now in Tamil, what we call that bye-bye as, right? That I know, Yepidi Rukinga. For hello, Vanakkam, Ipidi Rukinga, hi. So bye in Tamil, what we call bye. No EMK, not yet, will start it soon. In Tamil, Telugu I learned today. You have to tell me in Tamil. Let's focus on one thing first, na? Otherwise I'll mix up Tamil, Telugu, Kannada, I'll mix everything then. Chalo, theek hai, I'll learn it.